Now we have completed the kinetics. Okay, we have completed all the kinetics. We have completed general pharmacology. Now it's a part of dynamics. So all that was the kinetics. Okay, absorption, distribution, metabolism, elimination. All these are the process of kinetics. And last three processes: distribution, metabolism, and elimination. Drug deposition process. Now kinetics are over. Now we will see dynamics. We have started the dynamics. Now first, receptors. So what are the receptors? There are receptors on the your biological membrane for action of drug. Okay, so there are receptors. They receptors are nothing but they are the specific protein macromolecules. They are the protein macromolecules. They are present in the cell membrane. They are present in the cell membrane. They are present on the in the cytosol also. And these proteins will regulate the functioning of cell. These proteins or these receptors, they are involved in the regulation of functioning. कि वो कुछ काम रेगुलेट करते हैं इसका सेल्स का कैसा काम by altering the enzyme activity, by altering the permeability to ions, by making a conformational changes in or proteins. These kind of macromolecules which have functional correlate, they are known as a receptors. Okay, so they are nothing but the protein macromolecule of protein. They are present on the cell membrane. They are also present in the cytosol, and they are involved in the regulation of functioning of cells by altering the enzyme, either enzyme activity, either permeability to ions, or they will make a conformational changes to genetic material, or either they can alter the protein molecule. By altering or by changing these kind of things, they will act. Okay, so they are nothing but they are having functional correlates. That's why such a molecule with functional correlates, they are known as a receptor. हिंदी में बोलता हूँ रिसेप्टर क्या होते हैं? रिसेप्टर एक प्रोटीन के मॉलिक्यूल्स होते हैं जो जब ड्रग उनके साथ कंबाइन होता है तब वो सेल में जो भी सेल है उसमें चेंजेस करते हैं. कैसे चेंजेस करेंगे? उनका एंजाइम एक्टिविटी इंक्रीज डिक्रीज करेंगे, या फिर परमेबिलिटी ऑफ आयंस, जैसे सोडियम आयंस का परमेबिलिटी होगा उसको चेंज करेंगे, या फिर आपका जो डीएनए है उसको चेंज करेंगे, या फिर आपका जो प्रोटीन बनता है वो प्रोटीन बनने के प्रोसेस में चेंज करेंगे. वो जो फंक्शनल प्रोटीन्स होते हैं उनको बोलते हैं रिसेप्टर्स. ओके, so receptors are nothing but they are the protein macromolecules present on the cell and cytosol. Okay, and drugs will combine with this receptor, and then they will alter the functioning of cell by either enzyme activity, altering the enzyme, by increasing or decreasing the permeability, or by making a changes in the genetic material like DNA, RNA, or by making a change in the protein molecule. Proteinous molecule have a functional correlate. Okay, they have correlation with the function of cell. That's why they are known as the receptors. जैसे मान लीजिए कि अपने क्या है रिसेप्टर, मतलब टू रिसीव. क्या रिसीव कर रहा है रिसेप्टर? ड्रग को रिसीव कर रहा है. ड्रग को रिसीव कर रहा है. वहां पे स्टेशन पे खड़ा है एक जन, आपने भेजा है स्टेशन पे, कोई तो भी आने वाला है, उसको रिसीव करो, तो वहां से सही सलामत घर पे लेके आओ. दैट इज नथिंग बट दैट पर्सन इज नथिंग बट एक्टिंग एज अ रिसेप्टर. कि वो जो भी आपका गेस्ट है, वो गेस्ट बस स्टैंड पे आता है, वो पर्टिकुलर जो भी आपने जिसको बोला है आपके, वो लड़का उसको रिसीव कर रहा है, इसको आपके. सो वो लड़का will act as a receptors. Okay, so receptors will receive the drug molecule, alter the functioning of cell. Okay, so what is the nature of receptor? They are nothing but the protein macromolecules. Macro means large size. Okay, so they are large size protein molecules. Okay, they have functional correlates. So after drug will combining with the receptor, it will show either positive action or negative action by using either there will be alteration of enzyme activity, there will be a change in the permeability for the ions, there will be change in the genetic material, there will be change in the. So that's why these proteinous molecule have functional correlate, and that's why they are known as a. So there are different kind of receptors. We are not going to see in the details because it is, अगर आपको उसको पढ़ना है तो you can read from the reference. We are just going to see whatever is important for you. So ये there are different kind of receptors. Okay, there are majorly 4 classes: ionotropic receptor, metagotropic receptor, then kinase linked receptor, and then receptor that regulate the gene expression or intracellular receptor. Ionotropic receptor में क्या होगा? Any drug that will combine with the ionotropic receptor that will change the permeability to the ions, that will combine with the ionotropic receptor. These are the ion channels. For example, inside the cell there is a negativity. Cell के अंदर negative potential रहता है, -70 millivolt. मान लीजिए कि ये कार्डियक सेल है, it is a cardiac cell or it is a ion channel on the cardiac cell. So inside the cardiac cell there is negative, outside is the positive. When agonist will combine with this receptor, the permeability for sodium ion will change and sodium ion will enter in the cell. Okay, so here the potential is negative originally, but after the entering of sodium, the potential will change. There will be depolarization or activation of conductors, and it will show action. Such kind of receptors are known as your ion channels. Then second is nothing but the G protein coupled receptor. So these agonist will combine with this G protein coupled receptor. It will be activated these proteins. After activation, there will be generation of secondary messenger, and that secondary messenger will cause a cell signaling, either stimulation or inhibition. Then again, third category of your receptors is nothing but the kinase link receptor. Okay, so there is a tyrosine kinase. Tyrosine kinase is the enzyme that is linked with the receptors. That's why they are known as a kinase linked receptors. Okay, so agonist will combine with this receptor. It will cause a phosphorylation of tyrosine. Okay, and there will generate a signal, secondary molecule, and it will lead to activation of cell signal. Then again, last, that is known as a intracellular receptor, or receptor that regulates the gene transfer. Okay, so agonist will combine them. It will be transported across the nucleus, and there will be activation of transcription and translation process. Okay, so I have summarized all the receptors and whatever are important points from receptors here. So in this chart is very important. This chart is given in the book also. So I will explain this. So here in the first row you have type of receptors. Okay, type 1, type 2, type 3, type 4. There are 4 types of receptors. Is that clear? Okay, and in the column, first column, there what type of class is there, where they are located, what is a coupling, what are the example of receptors, what is the secondary messenger present in this. So they are given. So I will explain this one by one. Now let's come to the first type 1 receptor. So type 1 receptor, they are nothing but the ionotropic receptor or ion channel. Okay, as I have explained. So these receptors are known as ionotropic or ion channel receptors. Okay, so there will be either increase in the permeability for certain ions like sodium, calcium, or decrease in the permeability of certain ions. There is one ionotropic receptor known as a Gaba receptor A, Gaba A, beta subunit of Gaba A. That is the receptor, nothing but the benzodiazepine receptor. Okay, जो benzodiazepine का receptors होते हैं, जो benzodiazepine drug है जिसके ऊपर जाके act करते हैं, तो benzodiazepine drug will act on the benzodiazepine receptor, and it is a type of ionotropic receptor. So what is the role of that? So whenever any benzodiazepine drug will combine with that benzodiazepine receptor or GABA B receptor, that will increase the permeability for chloride ion channel. So there will be entry of chloride inside the cell. Already inside the cell there is negativity. Again chloride is electronegative, and it is also entering in the cell. There will be hyperpolarization of the cell. And hyperpolarization means inhibition of the cell functioning. Okay, so there will be inhibition. So that's why diazepam and all the benzodiazepine, they act as a sedative hypnotics. ठीक है, तो सेल का काम बंद हो जाता है, सुन उसके अपोजिट ही. सो फॉर एग्जांपल, अगर कोई ड्रग सोडियम की एंट्री बढ़ाता है चैनल में, जैसे आपका इफ यू सी cardiac potential, so normally what happens, so inside the cell electronegativity is there. Okay, inside the cell electronegativity is there. When there is entry of sodium inside the cell, there will be disturbance of your electronegativity. So there will be disturbance. So that is nothing but known as a depolarization. So whenever there is a depolarization, there will be the action potential, generation of action potential. There will be a positive action. Whenever there is a hyperpolarization, there will be a negative action. So diazepam will act as a depressant because it will increase the chloride ion channel, and there will be hyperpolarization of cell. So these type of receptors, they are known as a ionotropic or channel link. Okay, so they are located on the cell membrane. Okay, so what is the effect? Effect किस पे हो रहा है? So they are the nothing but the channels, potentials. Okay, so coupling, they are not coupling to anybody, they are direct. Okay, okay. So what are the example of this kind of receptor? So nicotinic acetylcholine receptor, then GABA receptor A, GABA A, जैसे मैंने आपको बोला है आपको इसका example. Then glycine receptors. So acetylcholine receptors, nicotinic particularly. There are 2 types of receptor for acetylcholine, muscarinic receptor and nicotinic. So muscarinic receptor are the G-protein coupled receptor. Nicotinic receptors are the ion channel receptor. In this case, there is no secondary messenger. But remember one thing: these ion channels will act in a millisecond, fraction of millisecond. So they are the fastest acting receptors. They are the fastest responding receptors. So these ion channels will respond in a millisecond. And this was the question in GPAT. So इसमें GPAT के लिए क्या important है? इसमें what is the class pulse? Class pulse is known as a ionotropic. Okay, and they will act in a millisecond. So second type of receptors. What are the second type? So second type of receptor, they are the Metagotropic receptor. Remember, they are the Metagotropic receptor or G protein coupled receptor. What we call them as a GPCR. So second type of receptor, they are known as a Metagotropic receptor or G protein coupled receptor. So okay, they are the seven transmembrane domain spanning protein. So they are heptahelical. So they are the heptamer. There will be 7 proteins spanning the transmembrane. So there will be 7 protein in the G protein coupled receptors. So it is a heptameric. उसमें कितना है 7 प्रोटीन रहता है, ये याद रखो हेप्टामेरिक. ये क्वेश्चन आया था एक बार. तो G protein coupled receptor are the heptameric. Then again G protein coupled receptor are also known as Metagotropic receptor. This was also again a question in GPAT. So this is the type second receptor. So they are also present on the cell membrane. Okay. Then again, what is the effector for these receptors? It is nothing but the enzyme, or they will couple with the G protein. Okay. Guanosine protein. Then what are the example of this kind of receptor? Muscarinic acetylcholine receptor, thyroid hormone receptor, then histamine 2 receptor, beta adrenoreceptors. All these are the examples of G protein coupled receptors. Okay. And in this case, G protein coupled receptor, there are secondary messengers. So CAMP, CGMP, calcium. All these are the secondary messengers. Then again, diacylglycerol is there. Then inositol phosphate is there. So secondary messenger के ऊपर भी क्वेश्चन आया था. So which one of the following is not a secondary messenger? So उसमें ऑप्शन थे CM, diacylglycerol. You should be clear about this. So what are the different secondary messengers? Okay. So secondary messenger के through act करेगा type second receptor. They are known as metagotropic or GPCR. Now type 3, they are the kinase linked receptor. So there is a tyrosine kinase enzyme. So these receptors are linked with the tyrosine kinase enzyme. There will be phosphorylation of them. Okay. So they are also located on the cell membrane. So they are coupled with the their effector is nothing but the enzyme. Okay. They are direct coupling. They will couple directly or indirectly with the enzyme. Okay, that is tyrosine kinase. Now examples: insulin receptors, growth hormone receptors, growth factor receptors, cytokinin receptor. All they are the kinase linked receptor. Okay. So in this case, what is secondary messenger? None. Okay, there is no secondary. In case of last type 4, that is nothing but the nuclear receptors or receptor acting on gene transcription. Obviously, as the name indicate, वो उनका presence कहाँ पे होगा? Obviously, cytosol में या फिर nucleus में. These kind of receptors are present in the cytosol or nucleus. Remember, these first 3 receptors are present on the cell membrane. Type 1, type 2, type 3. Means ionotropic, metagotropic, kinase link. These 3 are present on the cell membrane. These type 4 nuclear receptor, intracellular receptor, they are present in the cytosol or nucleus. And question might come: which of the following receptor is not a membrane receptor? So in that case, you should be able to answer. It is a type 4 or nuclear receptor or receptor that acts on a gene transcription. Is that clear? So what is the effector? What is the effect of these receptors? Gene transcription. Okay, they will couple to DNA. And examples of such kind of receptor are steroid receptor, thyroid receptor, hormone receptor, and again they will not have secondary messenger. Is that clear? This was all about the receptors. It actually is a big topic. ये थोड़ा सा vast topic है कि उनका signaling program कैसे होता है receptor का. अगर agonist combine होगा तो कैसे signaling होगा. That is another part. अगर वो part लेंगे तो मेरे में और आपके normal lectures में कोई भी फर्क नहीं रह जाएगा. Okay? So that is you have to read. If you are not understanding, you have to clear your concept from written book. एक book आता है हर एक topic के लिए अलग book refer. Now we'll see the next. Important terms to understand the drug receptor interaction. So your drug will interact with the receptor. Drug will show its action by interacting with the receptor. आपका drug receptor के साथ combine होगा, उसके बाद में ही action दिखेगा. Drug के direct action होते हैं लेकिन बहुत कम drug दिखाते हैं direct. जैसे आपका इरिटेशन हो गया, अगर इरिटेशन करना है आपको तो डायरेक्टली ड्रग इरिटेशन कर सकता है ना. इरिटेशन इज अ डायरेक्ट एक्शन. स्टिमुलेशन, डिप्रेशन, it is mediated by receptors. There are certain terms in relation with the drug receptor interaction. So what are these terms? These are nothing but the agonist, antagonist, partial agonist, inverse agonist. Okay, so I will explain these terms with the help of diagram. Then again I will explain in this slide. Okay, so first I would like to explain this with the help of diagram. So here is one diagram in front of you. Demonstration of agonist, partial agonist and antagonist and inverse agonist. So in this case, here is a y-axis. Okay, on y-axis there is a percentage response. Okay, receptor response, and on the x-axis there is the drug concentration. Is that clear? Now this is a basal activity. This blue dotted line known as a basal activity means zero activity, basal activity. Now suppose that this is your receptor, and these are your 4 drugs. Some drug is a full agonist, some drug is a partial agonist, some drug is a antagonist, and some is a inverse agonist. So green is a full agonist drug. Okay, light green is a partial agonist. Red, this pink is a antagonist. This brick red is a inverse agonist. Right? This is your receptor, basal act. Now, what is full agonist? A full agonist is a drug that will combine with the receptor, and will show full pharmacological activity. That is known as a full agonist. What is partial agonist? A partial agonist is a drug that will also combine with the receptor with the same affinity, but it will show less pharmacological activity. Affinity and efficacy. I will explain these 2 terms later on, but first understand these terms. Partial agonist will also combine with the receptor and will also show the pharmacological activity, but the pharmacological activity shown by partial agonist is less than the full agonist. Okay. Then next one is antagonist. What is the antagonist? Antagonist also will combine with the receptor, but it will not show pharmacological activity. Antagonist also will combine with the receptor, but it will not show pharmacological activity. Actually, it will block your receptor. Okay, it will cause blockage of receptor. So that is nothing but the antagonist. Now inverse agonist. So inverse agonist is also able to combine with the receptors, and it will also produce a pharmacological action, but this pharmacological action is opposite to that of full agonist. देखो यहाँ पे ये action opposite है उसका. Full agonist means a drug that will combine with the receptor and will show full pharmacological action. Partial agonist means what? A drug that will combine with the receptor will show partial pharmacological response or less pharmacological response. Then antagonist is a drug that will combine with the receptor, block the receptor. It will not show any pharmacological. Then inverse agonist is the drug that will combine with the receptor and will show pharmacological action in opposite direction of agonist. Right? Is that clear? Now let's come to the previous slide. What is agonist? So it is a drug which initiate or will produce the pharmacological action after binding to the receptor, and this agonist will have high affinity to the receptor. Affinity means ability to bind the receptor. Try to make a running notes file whatever I am saying, because sometimes what happens, I just say in the flow and that is not mentioned on the your slides. So it is a good habit to make a notes. Okay, so what is affinity? Affinity means the ability of drug to combine with the receptor. Okay, and what is intrinsic activity? Intrinsic activity means to show the pharmacological activity. Intrinsic activity means pharmacological activity. So agonist will bind to a receptor and will show pharmacological action, and it will have high affinity to the receptors, and it will also have intrinsic activity. So example, diazepam is an agonist on benzodiazepine receptors or Gaba receptor. Now partial agonist will also have equal affinity. It has equal ability to bind the receptor, but it has a less intrinsic activity. Okay, it has a equal or less affinity but less intrinsic activity. That means it will also bind to a receptor, but will produce pharmacological action but less than full agonist. Right? Example, pindolol is a beta blocker. Okay, so it is a partial agonist to the beta 1 receptor. Then pentazocine is a narcotic drug. It is also a partial agonist to the mu receptors. Just remember the example. इसके डिटेल में आपको चाहो क्योंकि अभी ये आपको हुआ भी नहीं होगा, जब आपको पढ़ाया जाएगा बाकी का तब आपको समझ में आएगा. Okay, so you have to just remember what is meant by partial agonist. So inverse agonist. So what will do? Inverse agonist will do. It can activate the receptor. It will combine with the receptor, but will produce action opposite to that of pure. जो action होगा वो opposite होगा. Okay. So example is beta carbonyl. Example is a beta carbolin, which is the inverse agonist to the benzodiazepine receptors. So beta carbolin drug है, so it is the inverse agonist to the benzodiazepine receptors. Normally benzodiazepine receptor का क्या रोल है? So जो भी agonist benzodiazepine receptor को combine होगा, that will hyperpolarize the membrane and that will cause depression, seen as depression, right? It is a normal agonist. Okay. Now what will inverse agonist do? So example beta carbolin. अगर बीटा कार्बोलिन बेंजोडाइजेपिन रिसेप्टर के साथ कंबाइन होके होगा तो उसके बाद में जो एक्शन मिलेगा वो क्या रहेगा? वो रहेगा स्टिमुलेशन. दैट इज अपोजिट एक्शन. सो बीटा कार्बोलिन इज अ इनवर्स एगोनिस्ट. Beta carbonyl is an inverse agonist. So that's why sometimes inverse agonist is also called as an antagonist. It is just a confusing terms. So what is meant by antagonist? Now it is also a drug that will combine with the receptor but will not produce a pharmacological action, and it will cause a blockage of your receptor. That is nothing but the antagonist. So antagonist will have same affinity like agonist but will have a poor or no intrinsic activity or pharmacological action. For example, atropine is a muscarinic blocker. Flumazenil is an antagonist to the benzodiazepine receptor. Atropine is a antagonist to the muscarinic receptor.